All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by my colleague from NCCAT, Ed Eng. Also with us from the Pacific Northwest Cryo-EM Center are Kirsten Wheeler and Sean Mulligan. And we're expecting Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2. He should be joining us shortly. Today's speaker is hosted by PNCC. Alex Vecchio is joining us from the University of Buffalo, and Sean will give him a proper introduction after a short update from each of the centers on the resources we make available at no cost to the research community. This webinar is a regular event we hold usually the last Thursday of each month, this month being our exception. And each month, center users generously share the tips and tricks that have enabled them to make use of Cryo-EM in their research with the help of National Center resources. So you can save the date already for our first talk of 2025. On January 30th, Rob Miller, currently in the Davis Lab at MIT, will share work from his Ph.D. in the Ando Lab at Cornell on the mechanism of nucleotide-dependent allosteric regulation in E. coli aspartate transcarbamylase. So please join us again next month. The registration link is already available on the events page at cryoemcenters.org, where you can also find recordings of our past talks. And finally, before we kick off today, Sean or Kirsten will be moderating Q&A at the end of the talk. So please use that Q&A box. Feel free to send questions at any time, but we'll hold them for the end. All right, so we can go ahead and get started. I will turn over first to Ed to let us know what's new at NCCAT.
Hi, I'm Ed, coming here from NCCAT. We have high-resolution data collection access, training and screening access on our 5 Krios instruments, 2 Glacios instruments, Tundra, and a whole host of devices. I want to highlight that in March and April, we're going to have our tomography short course. The deadline for applications is January 1st. So if you're interested, please go to nccat.nyspc.org.
Thank you, Ed. And we'll have to provide you a quick update from S2C2. So similar to the other centers, S2C2 has access to instrumentation for data collection as well as training. They have sort of regular working groups for their trainees with data processing and all of the information about their access categories and applying for time is at their website that you can see here. So check out S2C2. And Kirsten, I'll let you let us know what is new at PNCC.
Okay. Hi, my name is Kirsten Wheeler. I'm the administrative coordinator at the Pacific Northwest Cryo-EM Center. PNCC has 2 proposal types. First is the screening access proposal, which allows us up to 2 users to come on site for 5 days of sample preparation and screening with staff. We usually host 1 to 2 of these particular proposal types each month. The second type that we have is a general access, which is awarded up to 480 hours of microscope time a year for 2 years. The time can be utilized for screening as well as data collection. Approved general access proposals are eligible for training workshops, which cover topics from sample preparation through to data processing. We're excited about our recently added EPU data collection workshop, which we were successfully able to pull off 2 months ago, I believe it was now. Man, it's the holidays. PNCC has 6 microscopes. We've got an Arctica with a K3 camera for screening, as well as 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. Those Falcon 4i cameras are newer additions to our microscopes, and they are being overwhelmingly welcomed with open arms from our community, which is fantastic. 3 of our Cryos are equipped with Gatan energy filters, while the other 2 have Thermo Fisher energy filters. Our Cryos 1 has a Selectress X. The newest member of our microscope family, however, is a Glacios 2, which is still undergoing testing. We are hopeful to get that to us, hopefully by the beginning of next year. The upcoming training that we do have is Cryo-EM screening workshop. If you are a user of ours, you are able to apply for that. If you're interested in working with PNCC, I do suggest going to pncc.labworks.org and applying today. Our deadlines for proposals are the 1st of each month.
All right, thank you, Kirsten. I will stop sharing and turn it over to Sean to introduce today's speaker.
All right, good morning, everybody. It's my pleasure to introduce today's speaker, Dr. Alex Vecchio, one of our esteemed users from the Pacific Northwest Cryo-EM Center. Dr. Vecchio is an assistant professor at the Jacobs School of Medicine and Biomedical Sciences at the University of Buffalo. We've had the privilege of training Dr. Vecchio in his lab in cryo-EM, starting with his time at the University of Nebraska, Lincoln. Without any further ado, please join me in welcoming Dr. Alex Vecchio. Alex, the Zoom room is yours.
All right, I'll start sharing. Well, thank you, everybody, especially the PNCC folks, for the invitation. Hopefully people will be able to see this. All right. So, yeah, this is a story about someone who was working on a project who never thought cryo-EM would work for it, and then also found themselves in a place where we didn't really have cryo-EM infrastructure to begin with. So it's kind of a pep talk for people who are maybe in similar situations, that where there's a will, there's a way. So I'll start with the biology just very briefly, because to me, that's the driving force as a structural biologist. What we study is cellular junctions, and cellular junctions evolved during the first multicellular organisms, which are sponges. And then, the complexity of these junctions evolved based on tissue evolution. And so you can see over here, sorry, I have to minimize this. In vertebrates, that the complexity of the junctions is really sort of striking. And the ones that we study are these things called tight junctions. And the molecules that constitute tight junctions are called claudins. These are small membrane proteins, and they've actually evolved way back in sponges, but they never did the same functionality in anything until they reached vertebrates. And there's lots of reasons for that I won't get into. But what we're interested in is the macromolecular structure of tight junctions, which electron microscopy has sort of already enlightened us into, but what the proteins that constitute these junctions look like at the submolecular level, at the atomic level. And so you can see that 2 cells coming into close contact each other form these tight junctions, which are these very small spaces between cells. And then if you were able to rotate these cells 90 degrees, you'd see this sort of stitching pattern, the strand morphology that's present on one individual cell. And so, as I said, the proteins that drive tight junction structure are small membrane proteins called claudins. In humans, there's 27 subtypes, and their molecular masses range from about 22 or 23 kilodaltons to about 27 kilodaltons. They're fairly simple molecules when you look at them. They constitute 4 transmembrane helices and have a fairly small extracellular domain, which is derived from 5 antiparallel beta strands. Their intracellular domains are effectively non-structured. They have variable length C-termini that tether them to the cytoskeleton, and that's what helps junctions sort of form and deform under different epithelial and endothelial stresses. Now, we don't know very much about tight junction structure other than the structure of individual claudins. There are a lot of models, and MD has sort of shed light on how we think these proteins may assemble in both cis, which is on the same membrane, and in trans, which is across paracellular space. But they're just models, and sort of one of the goals of the lab is sort of to build the complexity of this of this system. But we've been interested in claudins for quite some time. Going in, when I started this project, we knew what the limitations were to try to get structures of these molecules. So it wasn't just that they were small. We also knew that the macromolecular assemblies dissociated in vitro fairly easily, and that was because we required detergents to solubilize them. And based on the molecular mass of most detergents that people use, they're very small in relation to the large size of the detergents. And effectively, what these large detergents do is inhibit crystallization because it basically obstructs protein-protein interactions. So we knew that these particular proteins had small extracellular masses and effectively no structured intracellular mass. So it was hard to build a lattice from this. And I sort of learned this the hard way by doing actual experimentations. The techniques themselves also had limitations. Crystallography, obviously, in high solvent content crystals and things like that, NMR for dynamics and size, and free detergents. But then cryo-EM, the molecules themselves at less than 25 kilodaltons are just too small to see, right? And so a brief history of claudin structural biology can really be encapsulated in one slide. This is all the claudin structures that have been determined prior to 2020. And what you can notice or appreciate here is that I have the claudin sort of colored all different colors, but there's very few structures, right? So the first one was published in 2014, and this was a lipidic cubic phase structure that was resolved to 2.4, but then all subsequent structures are much lower resolution. And what you can also notice is that all of them have some extra protein mass hanging off of them, right? And so what this protein mass is actually the C-terminal domain of this bacterial enterotoxin, which just biologically has been known prior to structural biology, at least, to target specific claudins. And researchers like me and others have used it in the high affinity binding that it represents to build type 2 crystals. And so the fact that we could add a 15 kilodalton protein mass on the extracellular side of a claudin allowed us to get type 2 crystals, and that sort of is how claudin structural biology exploded. But since then, crystallography has sort of stagnated because most of the claudins that bind this toxin with high affinity, the structures were determined. So we kind of had to come up with some new strategies, I would say. So just try to remember what resolutions we're talking about here when I start talking about this a little bit later. So we're not just interested in tight junctions and claudins per se. The fact that these molecules bound this toxin was interesting to us as well, because this toxin is a common gut bacteria. And then under certain conditions, it secretes this enterotoxin into the gut where it targets claudins and it binds the extracellular domain using the C-terminal domain. And then the N-terminal domain structurally rearranges in a way that we don't really understand to form this beta pore forming structure. And so the beta pore formed cytotoxic toxin is tethered to the membrane through claudin binding. A lot of these other beta pore forming toxins just bind lipids and form this pore, but this one requires claudins in order to do it. And just when we think about, therapeutics or biology or trying to get funding for these projects, we always considered that this toxin didn't have a vaccine against it, and it's like the third most prevalent form of food poisoning in the US. So claudin structural biology and understanding how this toxin works were kind of always coupled in our mind. So this brings us up to date with 2018 when I started my lab at University of Nebraska. So I started the claudin project in about June or July of 2012 in the Stroud lab at UCSF. And like all membrane protein structural biologists at the time, before cryo-EM really took off, crystallography was sort of the only game in town. And so I went through all the permutations that everyone who works on a membrane protein for the first time has to deal with, and trying to optimize the sample for structural biology. In about 2014, I started producing the enterotoxin and figured out which claudins bounded and which ones didn't. I got structures via X-ray crystallography of the toxins themselves. And then at the same time, I was setting up crystal trays of claudins alone and just not getting anywhere. So in about November of 2016, we actually sent our claudin toxin complexes for fab panning. And that's going to be a large portion of what I talk about today. But we didn't get crystals of these fab bound complexes. And so that was kind of abandoned. And then I got crystals of the claudin toxin complexes by normal vapor diffusion. And that kind of, was the major focus for several years because it took many, many, many crystals to yield structures. And that's kind of brings us to the point of when I started my lab. So when I started my lab, I knew that what took me 6 years in the Stroud lab to do was not sustainable for a new PI, and it definitely wasn't sustainable for new trainees. And so just in sum, it took me 5 to 9 years to get the 3 structures that I mentioned. That required about 80 constructs, 1,396 well plates, which equates to about 300,000 crystallization experiments, 16 eight-hour synchrotron trips within 9 months, screening about 100 crystals a session, right, to yield only 3 structures. And again, I knew that this wasn't sustainable long term. So back in 2017, before we even really made inroads on this, what we did was try to use these things called fabs to chaperone crystallization, which was what their major purpose was at the time. And so we collaborated with Tony Kozikoff's lab at the University of Chicago, and we sort of leveraged this claudin toxin binding interaction to pan for fabs. And so what we effectively sent him was human claudin 4 in a detergent micelle, and we formed a complex with the C-terminal domain of this toxin. The reason we did this was because we thought that claudins didn't have enough antigenic surface to find fabs against them alone. So we figured, let's try this. If we can get some fabs out of it, then maybe we can screen again without the toxin, okay? And so he has a fab phage display library that I won't get into. I kind of reference it down in the bottom. And if your input is some protein detergent complex or now nanodisc complexes, the output hopefully for you is some sort of fab. And so these are synthetic antigen binding fragments, the fab domain of antibody. And these are a two-chain antibody fragment with 6 CDR regions. And the CDR regions vary in sequence and structure, and those are what bind the target antigen. And so once we obtained fabs, the goal initially was just crystallization chaperones. But thinking big picture, and as the years went by, people were starting to use fabs as fiducial marks for cryo-EM. And, because they have these multifunctionalities and high affinity and selectivity, we always thought that they could be used for many, many different things. Most importantly for us was to enable claudin structural biology, but we also needed probes for in vitro and in vivo functionality because so little therapeutics targeted against claudins. So we thought that if we could find binders against claudins, we could get templates for therapeutics. And in the context of the toxin, we figured if we could find fabs that sort of bound a unique area of this complex, maybe we could prevent the cytotoxic pore from forming. So for us, the fabs were very exciting. So the output of the fab panning, we discovered essentially 3 fabs, which we termed COPs, which stand for CPE obstructing proteins. In hindsight, this wasn't a very good name, but when you're doing this, you kind of don't know what they'll be useful for. We very quickly, biochemically and biophysically, characterized what these fabs can and can't do. And so we figured that COP2 and COP3 actually share the same epitope, and they bind the C-terminal domain of the toxin here, shown in yellow. They bind it by itself. They bind the protein detergent, or they bind the claudin toxin complex, but they don't bind claudins alone. So we knew that it had a surface, a binding surface that was specific to the toxin. We also discovered this other fab, which we termed COP1, which we saw bound the toxin claudin complex, not the toxin itself, but did bind the claudin itself. So we thought that it most likely bound at an interface between the two proteins. And so this one immediately was very interesting to us, but we never obtained crystals of it. We did, however, obtain crystals of COP2 and COP3 bound to this complex. But like I said, the diffraction of those wasn't very reproducible. And so we kind of sat on this. We knew biochemically how they worked, but they weren't working for crystallography. But when I started my lab, I met with Kozikov lab, who said these fabs are now being used for cryo. Have you thought about using them for cryo? And I said, yes, I have thought about that, but we don't have any means to do this because the University of Nebraska didn't have any cryo-TEM capabilities. And I think around that time, the NIH centers were sort of still coming into development. Luckily for me, I had a former colleague start his lab at the Michigan State University named Ben Orlando, who during COVID, while he started his lab and was waiting for orders and supplies and people to start working on his own projects, they had a 200 kV Arctica at Michigan State. And so he said, do you have anything that you want me to put on a scope? And I said, yeah, sure. I have these fab-bound complexes and these toxin-bound complexes that we know are, some of them even crystallize, but we can't get any structural information from them. So we applied what we knew how to do well based on X-ray crystallography, which is make high-quality protein complexes for structural studies to Cryo-EM. So in the top panel, I'm just showing you some of the size exclusion profiles from some of these complexes. In the second group of panels, I'm showing you the negative stain done on the Arctica of some of these complexes in different detergents like LMNG, DDM, and even amphipoles. And then the negative stain 2D classes were so good that we were able to obtain 3D or high quality 2D classes and 3D reconstructions that were low resolution at the time to give us sort of the first glimpse of structural information of some of these complexes. And so the fact that this works so well in negative stain made us immediately want to go into cryo with it. I had no idea what that meant or how to do it, but Ben did. And so he took the protein that we prepped, vitrified it, put it on grids, and collected data on it. So I'll talk more specifically about the 3 boxed complexes. So this is Claudin-4 bound to the full-length CPE structure, bound to the C-terminal domain structure in a fab in either amphipoles or LMNG. And what I'll tell you is behind the scenes, we were also working on obtaining structures of the full-length toxin bound to claudins using X-ray crystallography. And essentially a year into starting my lab, we already had crystals. But just like every crystallography project of a membrane protein, initial diffraction started, I think, sub 20 angstrom. And it took us a ton of time and effort to try to optimize this. But no matter what we did, no matter how many crystals we screened or what sort of tips and tricks we did, we kind of plateaued at 7 angstroms. And we couldn't phase that data. Therefore, we couldn't generate maps from the 7 angstrom complex. So all we had was this predicted Claudin CPE structure prediction, which is based on our structure with the C-terminal domain, okay? Within a couple of weeks of sending the sample to Ben's lab and doing it in actual cryo, we were able to obtain 2D classes of the complex in a low-resolution 3D reconstruction at 13 angstroms. And, the maps aren't extraordinary by any means, but you have to appreciate that, since 2014, we knew that we could form these complexes, but we couldn't actually visualize what they looked like. So I was extremely frustrated. So even though this was a 13 angstrom map, it was still the first structural information we really were able to obtain of this complex. And so this gave us the idea that, yes, Cryo-EM is a potential way to go for this. The second and third data points that verified this came from the fab-bound complexes, which we put on the microscope shortly thereafter. And immediately what we saw, and this was done on a 200 kV Arctica with a Falcon 3. So these were kind of smallish data sets. And the Falcon 3 is not really sort of the best camera anymore. But the 2D reconstructions showed us secondary structural information. And the maps that we generated from this, even though they weren't atomic resolution by any means, allowed us to place within the map unequivocally all 3 proteins. And more importantly to us, in the 6 angstrom resolution map, we could see the little claudins hanging out within the detergent micelle. We couldn't really tell that they were a 4-helix bundle, I would say, but the fact that we could even resolve it with this camera, with this scope, with these sort of smallish data sets gave us tremendous hope. And so this really convinced us that cryo was something that we had to do. So now that we knew that we definitely had to learn cryo-EM, we had to think about the how. And so we lived in what I would call the Wild West of cryo-EM, which is effectively a desert. And the desert is now sort of more populated with cryo-EM microscopes, but at the time, late 2019 or early 2020, I don't think there was any cryo-EM microscope in this entire region. And so we had no access to cryo, and we couldn't just keep sending samples to Ben because he had to do his own thing. Luckily for us, Nebraska is what's termed an IDEA or an EPSCoR state, depending on if you're looking for NSF or NIH funded research. And so what that means is these states are traditionally underfunded from NIH and NSF, which means there's specific mechanisms if you live in this state to do high-end research, even though you may not have the infrastructure for it. And luckily for us, the NSF has such a mechanism. It's called an R2 Track 4. And effectively what this grant is if you live in an EPSCoR state, you can apply, you can write an idea, and they'll pay you to go to a national facility, learn some new sort of state-of-the-art technique, bring that information back to your university, and ideally start to train people in it. And so we wrote this grant, I think at the end of 2020 or early 2021. And luckily for us, we received this funding in January of 2022. And so this gave us the mechanism to do cryo-EM at one of the national centers, even being in a state that didn't have that infrastructure. And so what we planned to do was visit PNCC basically for 2 whole months every year for 2 years. And so in July of 2022, myself and a postdoc from my lab, Dr. Sirajan Kapoor, visited PNCC for the first time. And so we brought our own samples, and that's kind of all we had. And we learned and applied cryo-EM basically on the fly. So some of the complexes that we started with were these Fab-bound complexes because we knew that Fabs were important. We learned from the Arctica data essentially what COP2 and COP3 could tell us, but we were interested in COP1 because we already knew that it had higher potential as far as checking off some of the boxes that we thought Fabs could do for us. And so we dove headfirst into cryo-EM at PNCC for that first month. Like I said, we brought our own samples. We even prepped some samples there, and we screened everything that you would normally screen over the course of 3 to 6 months or a year. We did it, I think, within the whole month. By the end of the month, we kind of learned everything we needed to learn about these complexes. So we brought complexes in different detergents, and then we did all the things that people do. We tested different blot times and blot forces, ice thickness, different grid types, different hole sizes, different chemistries, and different manufacturers. And what I'm underlining here are all the things that we figured out worked best for our particular complexes. So our complexes liked longer blot times with very light blot forces because it favored what we'd call medium thickness ice. And what we realized that too thin of ice dissociated the protein complexes. And there was obviously air-water interface issues that all sort of proteins undergo. Another thing that we realized was the ice that our complexes favored the most were manifested themselves from small hole size, and that gold grids were far superior to copper grids. And specifically for us, the ultrafoil grids were superior to ultra AU flats. And on the right, I'm just showing you some of those small data sets we collected during our sort of screening month. So on the left you're seeing a gold flat data set, and on the right just a quantifoil copper data set. And this is effectively the same number of micrographs, the exact same protein prep with the same blot times blot forces and everything else. And we could unequivocally see that the contribution of the map to the micelle was minimized with the gold grids, and we could obviously see more secondary structure in the protein complexes with the gold grids than we could before. And so for us, this is critically important because we need resolution better than 4 angstroms to see the little 20 kilodalton membrane protein embedded in this large micelle. And so now that we knew exactly how to prepare samples and how to vitrify them and what grids we needed to use, we immediately sent fresh complexes to our collaborators at the University of Chicago and Tony Kozikoff's lab, who has access to a 300 kV Krios. And the first complexes that we sent them was the complex of Claudin-4 bound to the C-terminal domain of this toxin, bound to COP1 with an anti-Fab nanobody. And immediately we could tell that the data was going to go to high resolution based on the 2D class averages. And this resulting structure ended up to be 2.62 angstroms. And so on the bottom right here, I'm showing you each individual protein of this 5 protein complex. We now have the structure of COP1 bound to this complex, and it explained everything that we understood biochemically about it prior. In the top left, I'm showing you a local resolution map showing you how the resolution fluctuated across the structure. And for what you can appreciate, hopefully, is that there's areas where the Fab binds the claudin that exceed 2.4 angstroms, even though the global resolution was 2.6. And again, I want to take you back to claudin structural biology and have you appreciate that this structure, even though it was the first from Cryo-EM, immediately challenged X-ray crystallography for being the highest resolution structure. And so to us, this was incredible that we could obtain resolutions that were better than X-ray crystallography of a complex that we were interested in. The tertiary structure of the entire human Claudin-4 was visible in the maps. And again, this was significant to us because the poor resolution of some of the other Claudin structures and just the dynamics of the extracellular domain when it's not sort of clamped down by 2 molecules. In every previous Claudin structure, we couldn't really unequivocally assign every amino acid from the N-terminus to the end of TM4. But in these cryostructures, we could. And so the benefit of extra resolution or improved resolution allowed us to really model the entire tertiary structure of a claudin at the side chain level. When we looked at the structure, we were very satisfied because there were aspects of it that complemented X-ray crystallography. And that was important to us because we were always worried based on the X-ray structures because they were slightly low resolution and just the fact that the proteins have to pack in a lattice and there's protein-protein interactions that may affect how the protein complexes basically bind each other naturally. We were always worried that what the conclusions that we were making were sort of artifacts. But immediately when we looked at the maps and the protein interactions between the toxin here in yellow and the claudin in green, we saw the exact same contact protein interactions that we saw in X-ray crystallography. But importantly, Cryo-EM didn't just complement crystallography, it actually extended it. And what I mean by that is we were able to see things in the Cryo-EM maps that are impossible to see by X-ray crystallography, like the detergent micelle and also where detergents sort of bind the protein complex. And this is a factor of being able to visualize the micelle, yes, but also the improved resolution. And this became important for us because the micelle is a natural boundary between the soluble and the hydrophobic domains of the protein. And yes, you can look at the sequences and kind of predict how much of the protein is exposed and how much isn't. But being able to visualize the micelle allowed us to actually quantify what the soluble surface area of this protein is. And what we figured out by doing this was that only 10 kilodaltons of a claudin is actually exposed out of the hydrophobic environment. And that's really important to know when you think about trying to target these things with therapeutics. The maps were high resolution enough that we could even resolve a detergent molecule in between the Fab and the claudin binding domain. So this was the first proof that crystallography was sort of a dead end for some of this biology, and that cryo-EM was sort of the new thing that we should be focusing on. And so once we knew how the Fab worked and that we could get structures, we immediately went back to this claudin full-length toxin story, because at the time we were still trying to get crystals and we were still stuck at 7 angstrom. And we wanted to improve on this 13 angstrom structure that we were sitting on, because we didn't think we could effectively ever publish this structure. And so based on sort of an informal discussion while I was at PNCC with Craig Yoshioka, where I was saying, the Fabs, we know how they work and they're going to get us structures of a lot of these different things. He basically kind of quipped and said, have you ever just tried putting the two proteins together on a grid? And I said, I thought about it, but one's 35 kilodaltons, the other's 25 kilodaltons, and we kind of think that they're dynamic. So, why try to do that experiment when we know that the Fabs have a better chance of working? But because we had access to PNCC at this time, we basically, when I started my lab at UB in December of 2023, the first complex that we prepped was this claudin full-length toxin complex. And as a fail-safe, in case the claudin toxin alone didn't work, I also added the Fab into the purification. And so we did 2 independent purifications, one with Fab and one without. And again, this is just a negative stain from the data that Ben Orlando did years prior. And then we sent these complexes vitrified already, I believe, in Buffalo to Portland and collected data. And immediately, we could see secondary structure of both complexes. So we kind of had an idea that this would work. So this gave us the structures of the claudin full-length CPE complex by Cryo-EM. It's something that we've been working on for 4 to 5 years on for crystallography. The resolution of the claudin toxin complex without Fab, I would say the data processing was very challenging because of the small dynamic nature of this complex and the contribution specifically of the detergent micelle. And the data itself was just kind of not perfect. We did manage to get this down to 4 angstrom in CryoSpark, but to me, the maps are actually more like 5 or 6 angstroms. But it didn't really matter because we didn't need atomic level insights from this structure, because we were able to obtain that with the Fab-bound structure. So the Fab-bound structure was resolved to 2.8 angstroms. And the important thing here is, with or without Fab, when you superpose the yellow and green parts of each of these complexes, they effectively look identical. So any conclusions that we make based on the high-res structure can be applied to the one over here. And so what that meant to us was that the binding of the Fab didn't really affect any of the interactions that the claudins have with the enterotoxin. And this makes total sense, because if you remember how we panned for these Fabs, we used effectively this domain and the claudin itself. So the Fab should react to that complex rather than affect it in a given way. So let's bring you sort of up to date now based on what I talked about in 2018. So I told you that it took 5 to 9 years to get 3 structures via X-ray crystallography. Once we started doing cryo-EM, the first one came, even though it was lowest resolution, in about October 2020. And then once we learned cryo-EM from our limited access and then general access proposals at PNCC, and started to acquire this training by the NSF EPSCoR grant, the structures started coming basically anytime we wanted to put a complex on a grid. And recently in a grant, I wrote this, and I was kind of immediately amazed even though, we kind of understood this. I wrote in the grant that it took us 9 years to determine 3 structures by crystallography, but 3 years to determine 13 structures by cryo-EM. And, whoops, sorry. And that to me, it just hopefully highlights the power of cryo-EM, but more importantly, having a foundation and tools to enable cryo-EM. And so that's what I'll talk about now. What I hopefully convinced you of is that this combination of high quality biochemistry with fabs and a little bit of cryo-EM know-how totally and completely enabled Claudin structural biology for us. So not every Claudin its structure can be determined using these tools, but many of them can. And we also knew, based on some biophysical analyses, that the fabs could probe in vitro structure and function. But what we could have stopped there because we're structural biologists and kind of, sometimes that's all you care about. But we thought it was amazing that we had molecules that actually bound Claudins, knowing that only about 10 kilodaltons of surface is accessible. So we wanted to really see what else they're useful for. We wanted to see if they could probe in vivo functionalities, could be templates for therapeutics and or inhibit toxin like we kind of wanted them to do. And so one of the first things we did was we took this structure that I just showed you of, which is the fab-bound complex of full-length toxin bound to Claudin. And we kind of modeled what the progression of this complex would be into an actual cytotoxic pore, because none of these structures are determined. And none of them are possible in detergents because they require the membrane and or Claudin interactions to form the cytotoxic pore. But because we called these CPE obstructing proteins, we had an assay to test whether these fabs could actually obstruct cytotoxicity. And they could obstruct it a multitude of different ways. This fab, because we knew it kind of bound this interface, we didn't really think it would do anything to inhibit cytotoxicity, but we tested it anyway. And so what I'm showing you here is a cytotoxic assay where the cells on the top here are alive and the cells down here are dead. And these ones are dead because we've given them the CPE enterotoxin. And what we're looking at down here is no matter what version of toxin you give the cells, it kills them to a certain degree. But if you add this fab called COP1 into the experiment, you can see that, the magnitude of cell death isn't as much as with the toxin alone. So there is some partial obstruction of this sequence. It's just not very significant. But the fact that it did anything was kind of interesting. At the same time, we were working with Tony's lab to find new fabs. And so we found this one called COP4, which we specifically tried to find targeted against this full-length CPE. And because we were already schooled in the protein biochemistry and cryo-EM, when we received this fab from Tony's lab, we did our normal purification, we did our normal data collection, and we got a structure of it within 4 months. We got a structure of it before we even know what it did. And that's just because the platform was already so established. And so once we saw the maps and understood how this fab bound, we thought, okay, if these structures or these models are correct, this one might actually have obstructing potential. And so again, we tested it with the same exact assay. And so here's what GFP tagged Claudins look like normally in a cell. You can see that they're nice and green and the cells are alive. If you give these same cells enterotoxin, you can see that the enterotoxin kills the cells and the cell death basically digests and proteolyzes all the Claudin, and so you don't see fluorescence anymore. If you give cells this toxin and one of these other fabs that we worked on that doesn't do anything, yes, it also doesn't affect the cytotoxicity of this toxin. But COP4, we saw really strikingly that there's more fluorescently green cells. And what's amazing about this is in this particular assay, we only added enough COP4 where there was a 1-to-1 molar excess of the toxin. So we're talking about like 300 nanomolar concentration of fab, and we could unequivocally see cytotoxicity inhibition. And we just quantified that with live and dead cells. And you can see clearly the ones where COP4 is present, there are more live cells and less dead cells. And so this is another tool to get structures of claudins, yes, but more importantly, it's a tool to understand the biology. Because COP1 wasn't an obstructing protein, we wanted to figure out what else it could be good for. And so it was COP1 was discovered against, remember, human claudin 4 bound to the CPE cytotoxic domain. But because we have the capability to express and purify many different claudins, and we knew that COP1 bound claudin alone, what we wanted to see is what else can COP1 do? Is it really specific for human 4, or does it have sort of a broad claudin binding ability? And so we expressed a whole bunch, I think 13 human and mouse claudins, immobilized COP1 on biosensors and tested whether it could bind these claudins. And we saw unequivocally that COP1 could bind every single claudin tested. So we knew that we had discovered a pan-claudin binding molecule. And based on the structures, we knew where it bound. And so the next thing we did was this transepithelial electrical resistance assay, which basically measures the resistance on one side of a cell monolayer compared to the other. And what it is basically a proxy for tight junction barrier integrity. So high resistance means the barrier is intact, and low resistance and or obstructions to the resistance means the barrier is being perturbed. And so we use this tier assay to assess whether the C-terminal domain of toxin, the full-length toxin, COP1 and sort of COP2, which we knew only bound CPE, how it would affect transepithelial electrical resistance. The reason we use small toxin was because in the literature it has already been known to be a tight junction modulating protein, and we knew that the big toxin here, the full-length one, should kill epithelial cells. And sure enough, when we did the tier assay, that's what we saw. 100% tier means that the barrier is intact. And when we applied the small toxin to the basolateral and apical surfaces, we saw that the barrier integrity decreased. We saw the exact same thing with COP1, although the magnitudes change. With COP2, we saw no real effect on barrier integrity because it doesn't bind claudins specifically. And with the toxin, we saw that the cells were dead. So there was no effective tier. The barrier was completely obstructed. So this Fab, even though it was designed for structural purposes, is a tight junction modulating protein and can actually open tight junction barriers for potential therapeutic applications. And it does it in a concentration dependent way and in a reversible way. So this Fab is actually not just a probe, but also a modulator of tight junctions, which means in whatever oligomeric state claudins are in, when they're forming tight junctions, the epitope, the COP1 binding epitope is at least partially accessible. And so there might be tight junctions where this isn't the case, in which case COP1 should have no effect. So COP1 can be a proxy for us to understand what the structure of the oligomeric strand morphology is in a given tight junction. So in our hands, at least, these Fabs not just enabled claudin structural biology, but they were probes for both in vitro and in vivo function, potentially templates for therapeutics because they modulate tight junctions. And we also discovered one that inhibits CPE cytotoxicity. So hopefully this kind of brings home this idea that nothing is really intractable if you find the right tools for it. And obviously, if you find the right collaborators and the right infrastructure. So with that, I'd like to thank my funding, specifically in this case, the NSF for the EPSCoR funding, which allowed us to acquire expertise in Cryo-EM, and all the people at PNCC, the Orlando lab, and Tony Kozikoff's lab for providing us the tools and the expertise to take what we knew how to do and kind of reach the next level with it. So with that, I'll take any questions. Thanks.
Thank you so much, Alex. I'm I'm sure there's lots of applause. That was a bit overwhelming, the number of structures we saw there.
Yeah, it's a good problem to have.
Yeah, yeah. All right. I'm gonna let Sean take over with the Q&A.
Yeah, okay, so we have one question so far in the Q&A from Suresh Kumar. Hi, Alex, this is really nice work, especially dealing with low molecular weight proteins. Congratulations for that. I was wondering, that what's budding out in region of LC of Fab that can be seen in 2D and 3D? Thanks.
So I think that question, should I have my slides open? I'm not sure. I think that question is about the nanobody. So there's in one of the images, there's a little gray blob sticking on the light chain of the fab. And so that is a tool that was developed by another lab, that the Kozakov lab just kind of adds to every prep, because it locks down the hinge region between the constant and variable domains. And for most things, it's probably not required, depending on the size of the molecule at the other end of the fab. For ours, we haven't really tested whether it's good or bad. My opinion is that it's actually really important for our stuff, because it adds asymmetry to the complex, and it allows us to differentiate the heavy versus the light chain, and that sort of helps a lot in the particle alignments. So we can see it in 3D, and sometimes it's not perfectly resolved, but what we're really using it for is a tool to add asymmetry to the particles, I think.
Excellent. All right, we have another question. Thank you for the great talk. When you ran into issues resolving the membrane regions within the detergent micelle, were there any particular steps that you took that helped, or any tips you suggest?
So what I can tell you is the only time we can actually resolve the transmembrane domains within the micelle is if we unequivocally trap a claudin from both sides. So we need the Fab on one side and we need the toxin on the other side. In blocking the extracellular domain helps block the transmembrane domains, because when you think about how claudins react or work in vivo, their extracellular domains form all these interactions, and those interactions are transduced through the TMs into the intracellular domains, and then signal to intracellular proteins to bind or not bind, and be phosphorylated, not be phosphorylated. So for us, blocking the extracellular domain enabled resolution of the transmembrane domains. There's really no trick other than that. I would say collect a lot of data, because sometimes, even though the same complex might look the same in 2D, in 3D, you'll definitely see that the TM domains are sort of less resolved in some groups of particles.
Great. Another one about the Fab. What strategies were used to design a Fab specific to the claudin protein? How long did it take?
So all three of those Fabs were discovered with the same panning. So we sent one complex of claudin bound to the C-terminal domain, and all three were discovered simultaneously. There are probably 5 or 6 more that bound with lower affinity also present there, but they just didn't make it through the ELISAs. So I guess what I'm saying is however long it takes Tony's lab to do the the phage display, which I think is on the order of several weeks, that's how long it took.
Great. Another one here. Very interesting work. I'm wondering if you could define what you mean by medium thick ice. Do you have trouble obtaining sufficient contrast for these small complexes once the ice gets too thick?
Yes. Yes. So too thick of ice gives us contrast problems. Too thin of ice dissociates the complex. And so we need this kind of hybridized ice where the complex is intact, yet thin enough so that the signal to noise is good enough for us. I'm assuming every protein has these similar problems. What I can also tell you is our complexes do have sort of an orientation bias issue that we still haven't kind of gotten over. And that might be because the ice is still too thick for it to sort of organize in a true three-dimensional organization.
Any plans on capturing the toxin-induced pore structure? You mentioned detergent micelles would not work.
Yes.
Yeah.
The answer to that question is yes. We are going after that now that we know how we can't get the complex.
All right, this will be good. Has there been any thought in venturing into nanodiscs? Was working with micelles just easier than nanodiscs? Thanks for the amazing talk and work.
Yeah, so some of the stuff that we screened at PNCC, like we brought nanodisk incorporated complexes of these same ones that I showed you. We are not as skilled at making nanodisk complexes, I'll just admit to, but for us, the reason that we can't get very high quality membrane protein encapsulated nanodiscs is because we're trying to separate free nanodiscs from a nanodisk with a 25 kilodalton thing in it. And we just haven't found the right biochemical way to separate those two molecules. And the easy answer to that is, why don't you just, have a tag on the claudin and not on the nanodisk, and then you can pull the claudin-bound complexes down from the others? Yeah, that's a strategy, but we can't elute our proteins in imidazole. And so we're working on that. But needless to say, all the nanodisk incorporated stuff we've put on grids end up just being empty nanodisks.
Excellent. So this is kind of two questions that are very similar. So they want to know what the concentration of your samples when doing cryo and what detergent worked best. And furthermore, did detergent help remove the lipid nanoshell around the protein?
So we don't see a lipid nanoshell around the protein, but that is almost certainly because we are exchanging our proteins from DDM, which they're solubilized in, into LMNG. So the answer to the one question is, for us at least, LMNG has been the best detergent. That's not to say DDM cholesterol or DDM itself or GDN won't work. We just haven't tried to reinvent the wheel. Because LMNG is working for everything. That's just what we're sticking to. That might be something that we look into further. What was the other portion of that question?
It was around the concentration, what detergent
Oh, concentration of protein, right.
Yep.
So these are 5 protein complexes. And so what has been working for us lately is I would say 5 to 8 mg per mil. They don't seem to have a problem sticking to grids. We just need a lot of particles to get all the orientations. And a lot of them tend to get washed away with the blotting paper. So we've tried back blotting, front blotting to try to optimize these things, but just hitting it with a high protein concentration for us has been good, and we haven't tried much less.
Okay, this one is about
Sean, can I pop in before we get to the next question? Alex, can you stay for another couple minutes to get to more questions? Awesome.
Yes.
So we're gonna stick around for a few more minutes to get to more questions. If you do have to head out at the hour, we're still recording. So you can come back to check the last of the questions when we get that recording up. And if you do have to go, thanks for joining us. Come back again for additional tricks and tips in coming months. And we'll let you guys get back to the Q&A. It's exciting.
Thank you. Yeah. So this one, another about the concentration. So characterizing the, is there any way to characterize the pore formation in intermediate states by concentrating the sample? Do they allow for you to, do the membrane-like systems allow for you to observe it?
No. So, I mean, we haven't exhausted all of these based on our modeling and modeling by another lab. It makes total sense to us now why when you have the claudin and the full-length toxin bound in a detergent micelle, why we can't form those complexes. Now, granted, we haven't tried 20 or 40 mg per mil, but you have to you have to understand that the concentration at which this toxin is actually cytotoxic is like 30 nanomolar. So potentially we could skew that equilibrium to form pores at super high concentrations. But the real reason it's not working is because the detergents are obstructing the interactions that are required. So it gets back to what I said on slide 3. I think it was detergents actually isolate only monomers because they break apart the natural oligomeric states that these proteins want to form in a membrane. So this is where we want to go as a lab, is to build on this complexity now that we kind of know what detergents can and can't do.
Great. So we have 3 more questions. One of them's on the sample preparation side, and the other 2 are on processing. So we'll go to this sample preparation one. So they recently discovered that quantifoil is having huge problems with producing 0.61 grids. Do you have any comment on that? Are you experiencing
It
it?
sounds like it sounds like everyone is sad about that. Yeah, I mean, when I started my lab here in last fall, I ordered 0.61s and I still haven't received them. So many of the structures that I showed in those are done on 1, 2, 1, 3 grids. And this is, I don't know if this is empirical at the moment, but basically we haven't been able to get past 3 angstrom with the 1, 2, 1, 3s yet. We're kind of stuck in the 3.2, 3.4 range. And I don't think it's the sample prep, and I don't think it's the ice per se. Well, it is the ice because of the small hole. So I can't say unequivocally yet that that's the case, but I have a feeling that once we get some 0.61s, we'll get back to sub 3.
Excellent. So this one is a beautiful story. Thank you for sharing. Do you have any tips and tricks for processing cryo-EM data of low molecular weight protein samples, such as the complex without Fab?
Oh, so let me think. I am very much still a novice in the cryo-EM processing. But basically anything that's mentioned in the CryoSpark blog posts or questions, I just follow those to a T. Because if you Google sort of membrane protein problems, or they have particular websites or links to the challenges in processing membrane proteins, I basically just follow what CryoSpark suggests. So many cycles, um, you know, nothing very unique, I would say.
Okay, excellent. And just this last one is asking about the impact of box size, and if the larger box size includes more high resolution signal, if you saw anything there.
So one thing we do at the end of our processing is once we have a bunch of particles that we like, is we go back and always re-extract them with no binning back to every sort of box size within that particular range. And we then do NU refinement at the end. And we basically end up picking the box size that ends up with the highest resolution. And so we don't stick with one at the beginning and assume it's the best. We always go back and sort of systematically test every box size, plus or minus, I would say, 100 picks from where we started. So computationally, we kind of brute force it, I would say.
Great. Yeah, thank you for answering those questions, and thank you for the wonderful talk. I think
Can I ask one quick question? I'm curious, now that you have these EM structures, can you go back and phase that 7 angstrom X-ray data? Have you tried?
Well, so the reason we can't phase that structure isn't because we don't have a good model. It's because the data is probably very ambiguous. Like we think it's in a certain space group, but it might not be.
Okay.
And that's because the resolution's poor.
That was my only question.
Yeah.
Thanks.
and so Christina, do we have any sign off?
Um,
Or
no, that's
okay.
it. So, Yeah, thank you, Alex, and thanks for hanging out to finish the rest of those questions.
Yeah, thanks everybody.
Thank you.